So let's start. In the last class, good afternoon, everyone. In the last class, we have discussed about 3 blotting techniques, and our goal was to uh, not just learn the principle, but also to be in a state where we can interpret the results. And this has to do with mutations, and we are trying to determine what kind of mutations are possible and how one can use the techniques of blotting. I think I asked you to practice this one or try to see if you can get through that. How many of you were able to solve it? Or you must be having some kind of
Sir, I have a doubt regarding the
some
fourth
answers
line.
at least.
Fourth line, sir.
You have a doubt regarding
The fourth line in the western blot.
uh, fourth
Yes,
lane.
sir.
Okay, that's right. The rest of it is the rest
It's
done?
fine, sir.
I was expecting that you will upload. Anybody will upload,
I will
but
do
I
it
don't
now.
think
I will do it
it,
now.
that's okay. Yes, you should do that though.
Okay, sir.
By now, if you have done, it would have been at least. Thanks for doing it. Anybody else has tried to do this? Anybody? Okay. I will try to solve it, and you can you can agree or disagree based on whether my reasoning is right or wrong. Okay. So first one is, as well, we said it is normal in all the cases. So we'll just write normal in all these cases. Subjects 2, 3, 4, 5 are kind of mutations. And by trying to compare with the normal and compare between the southern, northern, and western blots, we are supposed to make a qualified or educated guess about the nature of mutation. And that is what we will do here. So I will just take 2, and 2 is heterozygous. For this is assume this is for gene X. Okay, normal is XX, homozygous for, okay, not X, I should use something else. I'll use GG. Okay, normal is homozygous for GG. And person 2, he's having one of it must be G, and because there is only one band is corresponding to that of the normal one, whereas the other band, as is here, is having a larger fragment, and we don't know what it is, so it must be a different allele, right? So I'll call that as G1. Okay, and you can see that in from the northern blot, it is apparent that somehow for some reason G1 does not have a um, transcription. So person 2 in this allele, because one would have expected, you see that there is half the transcription, which means G1 has no transcription no transcription. This is also confirmed from the western blot here, where you see half the product of in person 2, but you don't see any other fragment anywhere, which may mean that it is a there is no translation, also no transcription, therefore there will also be no translation, right? So the mutation in G1 must have been in the promoter region. It could also be not just the promoter region. It all depends on the molecular biology of the gene. Say if this was a promoter, if this was a promoter region, which would have required an activator to allow its transcription. And the gene for this activator Okay, this is another gene coding for the activator gene. If this activator gene is mutated, then there is no production of activator, because of which there might not be a transcription in this. That is possible, but one should look at this region, this band here. There is a second band in southern blots, which shows that it has increased size, which would mean that most likely it is a kind of insertion within that region, and the insertion may have occurred mostly in the promoter region. That could be the possible answer for it. For patient or the person 3, you see that there is no normal band here. Okay, the band corresponding to the normal, it is not there. And the top band, which is corresponding to that of G1, which we call this is G, and we call this as G1. It is twice the intense, which would mean that this might be G1, but I said might be, and you can compare it with that of the northern blotting. You see the same phenotype, in the sense there is no transcription at all, which would mean, and also you would see that there is no protein band at all. So uh, person 3 is homozygous for G1. So it is G1. Person 2 is heterozygous. So I'll look at the other bands. Now I will look at fourth one and fifth one are a little bit complicated. Fourth one is having the same band as that of G, and the transcription is also, the mRNA size is also similar to that of G. But what happens is there is something wrong with this one. This protein is shorter in length, right? That's why it was traveling much farther, which could be um, that we will try to theorize what may have happened. It is being detected by Western blot, so the epitope is present on the protein, and the transcription, the size of it is normal. Okay, gene size is normal. That is, I'm determining that from the southern blot. From the northern blot, I am determining that the mRNA length is also small. So there are no major insertions or deletions. Okay, the size is, the genetic size is almost same. What may have occurred could be point mutations, it could be small insertions or deletions, or it could be frame shift mutations, or it could be nonsense mutation. Say, for example, there is a nonsense mutation. There must have, if a nonsense mutation occurred here, then you wouldn't be able to find out. Then the protein produced, this is the stop codon. Original stop codon should have been here, but now this is a new stop codon because of nonsense mutation. Therefore, the protein produced is actually smaller than which would have been a full length. It's a kind of truncated protein. So this is a new allele. We should call that as, we will call that as G2. And you don't see anything else. And the protein intensity, band intensity here, mRNA intensity is equal to that of the normal one here. The protein intensity is also similar to that of the normal one. So most likely 4 is homozygous for G2. And again, as I said, it could be a nonsense mutation within the reading frame, or it could be a frame shift mutation, which might have resulted in a smaller protein. Okay, those are the two possibilities. Otherwise, there are no big or large scale changes in the gene. but the effect is very evident in the at the level of protein. The same can be applicable, or same can be said about the on 5. Again, we should always look into the genome. The southern blotting, DNA fragment size is similar. Intensity doesn't matter here. Fragment size is similar. And in northern blot, you have the mRNA same size and intensity. So it is transcription is not the problem. The problem is with the fragment, with the protein. Protein is slightly larger. In that case, there are two possibilities, at least two possibilities. Oops, sorry. At least two possibilities. One of it is we will say if there is a stop codon here, say this is a TGA, right? Now it is resulting in a larger protein. Okay. And TGA could become into, say, GGA. Then when the mRNA is produced, upon translation, ribosome will start binding, will start initiating, will translate past, because now TGA is a stop codon is mutated to a sense codon. Okay, because of which it will translate until the next stop codon comes, whichever. That means the protein produced is larger than the normal size, because of which it is traveling slower, or its mobility is less than that of the other ones, normal one. So there is a possibility that the stop codon may have attained became a sense codon, and because of which it might have gone further. You have a question,
Yes, sir.
Madhumati?
Yes,
Madhumita, yeah.
sir. But it shouldn't be reflecting in the mRNA while it transcribes, sir.
What should it be reflecting?
The TGA converted into GGA.
Yes. Size of mRNA, do you think size of mRNA would change?
Depending on the extent, it would go and
No.
transcribe.
Transcription start happens because of promoter. I mean, it starts at promoter and ends with the terminator. Okay. So whatever happens in between, substitutions or any point mutations, it is not going to affect the length of the mRNA. Okay. The reading frame is from here. The ORF is from here till here. So the ribosome will bind here and will stop here. If there was a mutation, like I was saying, TGA mutated to GGA, then ribosome, now the new ORF would be until wherever there is a stop codon here. Say there is a stop codon here. Now ribosome will bind here and will translate until the stop codon. Is that understood? The mRNA size doesn't have to change in that.
Okay, sir.
Okay.
Okay, sir.
Uh, yeah, that's fine. That's good. As with the case in the previous one, the stop codon changing into a sense codon is a possibility, but also you may have a frame shift mutation somewhere here, because of which the stop codon is no longer stop codon because the reading frame has changed, and it will translate until a stop codon is encountered, and therefore you may have a bigger protein. So subject 5 is homozygous for, we are calling this a new allele, G3G3. Okay. If your answers are somewhere here, you are most likely right. You may have different other ideas also on this. Nothing wrong, but ensure you discuss with friends, and you can also defend your answers. Okay. So I will now move on to a topic that I wanted to discuss about sequencing. I think I must have asked you to find out what is bisulfite sequencing. If not, I'm trying to remind you again. Bisulfite sequencing. This is to do with sequencing to find out methylation status of DNA. Okay, when we were learning about yeah, we were using about epigenetic status during different developmental states. At that time, I must have asked you to find out about it. In today's class, we will discuss about two things, two techniques about sequencing. Now, they might not matter as much, but it is important for the way of thinking. Okay, that's why I wouldn't skip this. At one point, DNA sequencing was very hot in the sense everybody was trying to figure out the sequence. People have, after the discovery of DNA, the next thing people wanted to know is how the sequence is giving rise to a protein or so, how genes are there, and what is the genetic information, and so on. To do that, one should be able to find the order of bases in a DNA. And for that, there was a race between several groups. Two groups, one of it is Maxim Gilbert, and the other one is Sanger's group. Uh, Maxim and Gilbert's method are this method is called as chemical cleavage method, and I mean it is the principle is depending on cutting the DNA on the basis of what kind of base is present. That is followed by electrophoresis and detection. This is non-enzymatic method, and it is also referred to as cleavage method. So in this, while we try to read this, we first want to know about some things. The required material is single-stranded DNA. How can we get single-stranded DNA? That is where we were discussing about vectors, and we also discussed about single-strand vectors such as M13 phage and so on. So this is one of the application for why we should have single-strand vectors. So you need to obtain a single-strand DNA and then perform end labeling. The second one is end labeling. I think we discussed about random labeling. We didn't discuss about end labeling. If you want to distinguish both, there are two labels. This is a DNA molecule. If you want label to be anywhere like this in all over the DNA, just all anywhere you wanted it, that is random labeling. If you are looking to label only on one side, one end, that is called as end labeling. So I quickly have to ensure that you remember, recollect end labeling and first random labeling and then end labeling. In random labeling, what are the substrates? We use I will write this, sorry. Oops. In random labeling, I only want to talk about the substrates that we used. We would have needed a template. It is based on polymerization. So we need almost everything that is required for polymerization, including the enzymes. And in that, we use radio label. So one of the bases, say we are doing in vitro transcription, then we would have used NTPs. And one of the bases should be CTP, in which the phosphate is radio labeled. And we have three different phosphate groups, right? So assume this is the base. Then we have the sugar, ribose. And then we have phosphates. So this is alpha, beta, and gamma. And during polymerization, these two pyrophosphate is broken and released away. And this alpha phosphate, that is along with the alpha phosphate, this gets incorporated. Therefore, if you are using substrate NTPs, the alpha phosphate should have been labeled. So alpha P32 CTP. Okay. So during polymerization, DNA polymerase or RNA polymerase, as the case may be, it will remove this pyrophosphate, and this alpha phosphate will become the part of the uh, sugar phosphate backbone of DNA or RNA, whichever is the case. In end labeling, we are going to do it a little differently. In end labeling, we are supposed to only add a phosphate group on the 5' side. So one phosphate. What we need is the double-stranded or single-stranded DNA, whichever is the case, which you want to the target to be labeled. Then you need an enzyme. Can anybody recollect which enzyme should be used? What is that? PNK, polynucleotide kinase. Okay, that is the enzyme that transfers a uh, phosphate group from ATP to the substrate DNA molecule. So you will need ATP, and here we want this phosphate, the end phosphate, to be radio labeled. And ATP has 3 phosphates, alpha, beta, and gamma. The gamma phosphate is the one that is going to be transferred by PNK from ATP to the target DNA sequence, to the 5' ends. Okay. So we should have gamma P32 ATP. Okay. So I hope you understood that. One question, important question to ask is, when do we choose to have random label? When do we choose to have end labeling? Okay. Whenever you have the um, something you want to detect is in very, very minute quantity, then people will choose random labeling. So if you are doing southern blotting, for example, out of large amounts of DNA, a small proportion of it would be the gene of interest, and you're trying to detect it. A probe should hybridize, and the probability of finding it it becomes difficult. In that scenario, you want amplified signal to notify you that notify you the presence of the gene. In that, because you need to amplify that signal a lot, you would choose random labeling, because as you can notice here, there are multiple radio labeled atoms, you can say, right? So you have more atoms per hybridization in that case. So whenever you have the sequence of interest is very small, very low in concentration, it's like a needle in a haystack, then one uses random labeling, sorry. End labeling is when you have a large quantity of the molecule that you're working. This is, for example, the gene of interest. I already amplified it using PCR. I have a large quantity of it. I don't need to radio label it. Then it will give too much of signal, and also, why should one be exposed to radioactivity as much? We have to minimize as much radioactivity usage in our work, and exposure is not a great thing. So it's important that when we have too much of sample, we should have we employ end labeling. Okay, I hope you will remember these two things. So what I'm going to now discuss is about the technique of maximum Gilbert method of sequencing. So the first thing, I already described single-stranded DNA. Then we have described about the end labeling. We are going to use gamma P32 labeled ATP and polynucleotide kinase to add the radio labeled phosphate to the 5' end of the single strand that we have. Here is the single strand. Now this implies that it is radio labeled. Next important things in this method is cleavage at specific nucleotides. Remember, this is the cleavage type, or what the name is called cleavage method, or it is also non-enzymatic method because it's opposed to the Sanger's method. So there are four different types of reactions chemists have come up with, where one is G specific. That means if you have a sequence something like this, so G reaction means it will cut on the 3' side of the G, something like that. Okay, I think in this figure it is shown as 5' side. I remember it as 3' side. We will have a look at it later, but the logic remains the same. Uh, and the second reaction is purine, right? It could be either A or G. These are a set of reactions. I don't think the chemistry is important for us anyway. We don't use this, but for the logic sake, I wanted to take. So we will focus on the logic. And there is another reaction that is pyrimidine specific. Wherever there is pyrimidine, there will be a cleavage. Like you see in this reaction, C is there. There are T, T, C, T, T, and CC. So you may have a cut. You will take several, say hundreds of this single strand, and you perform pyrimidine specific reaction. So, and it is not complete. Okay, it's kind of partial degradation. So in some molecules, a cut might be made here. So you might have this. In some reactions, a cut might be made here, and so on. You will have different lengths of DNA molecules. Okay. And the other reaction is C. So if you want to remember it, you should remember it as G purine pyrimidine C, and try to make a kind of rhyme or something so that you can remember that. Yes. So I will take this sequence and I will try to perform what will happen in this case. I have assumed that this has been labeled in this case, right? You also, when the cleavage has occurred, there must have been the other part of the DNA here, this much part, and so on. But when you run the gel and perform radiography, autoradiography, what you will see is you will only see the bands that have the label. Remember that we have chosen a single strand DNA, and then you labeled it, right? And although you may have a fragment like this is the labeled one. If a cut has been made here, and you run the gel, you would actually, if it is just agarose electrophoresis, you might probably see a band like this, one which would have the label, and the other one may run farther. But upon blotting and autoradiography, you'll only see this band. You will not be able to visualize the other band because the other band there is no radioactivity to this one. End labeling is only on the 5' side. Okay. So you'll have a series of bands here. Like this one, it has 3 bases, so it will come to, in this case, like this. If you have ATT, GA, this is the second next band, about 9 bases, and the other one is a full length one. How we determine it is important. Okay. Here is the scenario. I hope you can help me out with this. So I want you to compare this position. In this position, what could it be? This lane would tell you that it could be either A or G. And what does this lack of band here, what does it say? It is not G. One is saying that it is A or G, and the other one is telling you that it is not G. So it must be, what should it be? It should be A. Look at the same thing in this position, which is say I'll call it as 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, and say 14. We will not go until there, just for there is little bit confusion there. But I hope you are able to see that from one lane, from the second lane, lane 1, lane 2, lane 3, and lane 4. Lane 2 will tell you if there is a band, it means it is either A or G. Lane 1 will tell you if it is G, right? If it is A, A and G is present, it is saying A and G, and lane 1 at this position it's telling it's not G, so it must be A. Lane 3 also a similar situation, same as this, so we don't have to go to that. We'll look at lane position 9. In this one is telling you that it is A or G, and the lane 1 is telling you that it is G, so it must be G, right? Same way you are going to do the other ones. The similar logic, the only reason why I have taken this Maxim Gilbert included is about how you deduce results. It is deduction, pure deduction, and a good way of thinking as well. Look at lane 4 and 5. In this is the position 2. What could it be? C and T. Pyramid in lane 4 is telling you it is either C or T. And lane 3 is telling you it is C. So in that position, it must be C. The same is the case with position 4. Let's look at position 5. This particular band is telling you it is C or T. And lane 3 in this location is not showing a band, which means it is not C. Therefore, it must be T. Using such logic, one has to determine the sequence. Let's see if we made it right. Yeah, so ACACT is right in this position, G is right, and so on. So try to have a look at it, and we can discuss this once again later. We will do an exercise on this, which will make it much more clear to everyone. The next one is called as Sanger's method. I think most of you are familiar with this. It is, I think he received Nobel Prize for, one of the Nobel Prize is for Sanger's method, I think. It is also called as dideoxy method, or the enzymatic method, or chain termination method. It's good to remember these names, because whenever there is in exams, competitive examinations especially, they might ask in any of these terms, and if we didn't know them, we might miss on that. So it's okay they're easy to remember. You use a dideoxy nucleotide, that's why it is dideoxy method, as opposed to the chemical degradation method of Maxim and Gilbert. This one involves enzymes, that's why it is enzymatic method. Because we are using dideoxy method, the polymerization is halted or chain is terminated, because of which you call it as chain termination method. So I hope you remember one of the questions does often come is about the chemistry of nucleotides. You remember ribose to start with. You should know ribose. Say something like this. This would be 1, 2, 3, 4, and 5 CH2OH. So I didn't put prime because this is not with the base. If it were with a base, I should have put 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. So ribose would have OH in second position, 2 prime, and 3 prime positions. Whereas deoxyribose, that is the sugar molecule in DNA, would not have a hydroxyl group at on the 2 prime carbon. Okay. Now the dideoxy here, the name is also good. 2' deoxyribose. That means in this one you do not have OH. In the case of ribose, there is a OH in the 2' on the 2' carbon. Dideoxy ribose is where both 2' and 3' carbons do not have OH. So I hope you remember that 3' OH is very important or is essential for all nucleotide polymerization, be it in transcription or transcription, replication, or even using terminal transferase. Anytime a nucleotide is added in biological systems, okay, chemically many things can be done in the lab, in vitro. That's a different issue. In biological systems, all polymerization or addition of a nucleotide onto a growing chain happens through 3' OH. That's why without 3' OH, there is no polymerization. So dideoxy in 2' 3' dideoxyribose, there is no OH. So that is used uh, as a one of the bases, so that chain termination happens. And the way people have conceptualized this scheme is also intelligent. It's very smart. um, I want to always emphasize on the way you can make a thought experiment, or think of imagine what all how one should follow the sequence of steps. So probably it must have been many people involved in inventing a technique like this, or maximum Gilbert too. That is logical thinking. So I would want that you people also always try to do that. So the 3' OH will make a nucleophilic attack on the alpha carbon, and that is how polymerization occurs. That's normal. But if there were a 2 dideoxy nucleotide, it will get incorporated, because the 3' OH of the primer would actually make the nucleophilic attack. It gets incorporated. That's fine. But now there is no more. Once the dideoxy nucleotide is incorporated, there is no OH. So there is nothing to make a nucleophilic attack and add a nucleotide. So the chain cannot be polymerized anymore. So the Sanger's method involves this ingenious idea of how we can use it to read through the sequence. So they have formulated the whole scheme is using 4 different reactions. So in which they will add all the DNTP polymerase, everything in all the reactions, 4 different reactions. But in one, they would have added dideoxy ATP. Okay. In the second one, they might have added dideoxy CTP, dideoxy TTP, and dideoxy GTP. So the rest of the compounds or buffers, enzyme, everything is common in all the other in all the reactions. Only difference is that in addition to them, they have in one reaction they would have added dideoxy ATP, in other dideoxy CTP, and in one dideoxy TTP, and the last one dideoxy GTP. Just let's look at what would happen for dideoxy in reaction 1 here. So the primer, this is you will need it. One will need a template and a primer, and the primer should be end labeled. Okay, remember that primer should have been, you take the primer and you use polynucleotide kinase and end label it with the gamma ATP, gamma p32 ATP, and then you allow the reaction to happen, polymerase chain reaction. So the primer may start with G. In anywhere there is T, okay, in the template there is T, then there is a possibility for addition of A. And whenever there should be an addition of A, there are 2 different types of A in this particular reaction. There is normal ATP, that is DATP, And there is also an additional added dideoxy ATP. So there is a chance that a normal one could get added or a dideoxy one could have added. If a dideoxy one is added, and that molecule will no longer polymerize, it's over, right? Chain is terminated there. But if a normal one has incorporated into the position, then it will proceed further reactions until another position where another A could be added, adenine. Again, the same thing. Normal would the normal one get added, or a dideoxy one could get added. If a dideoxy one is added, chain is terminated there. So in this way, the whole there will be a multitude of fragments with different sizes, and the sizes are based on the occurrence of A, right? Because we are now talking only about the reaction 1, in which we have added dideoxy ATP. So there will be different fragments. Each of them have varying in length, and all of them would have had a dideoxy at their end like this. Okay. So like that, in the 4 reactions, one is for ATP, adenine, other one for cytosine, one for thymine, and then the last one for guanine. And then one will run the gel, and then, as I said, as you can see here, they're all of different sizes. That means they would have traveled at different lengths. So when we are doing electrophoresis for these kinds of experiments, we should use high polyacrylamide gels. And then it should be denaturing. Denaturing in here is, since we should prevent any intramolecular hydrogen bonding. We will discuss about it when we'll talk about a technique called SSCP. We'll discuss there. So if you have A reaction when you load, and G, C, and T, each of the fragments will run at different lengths. They will run different distances based on their size, and each of these may have differences by 1 base, right? So when we read here is G, the base at that location would have been G, and in the second position it is A. Just we look at this and we can figure it out what is the base there. So T, C, C, like this. This is the primer. Okay, this is not the template. This is the primer. Before you have any more doubts, I would in the next class we are going to do a problem on this, on sequencing both Maxman-Gilbert method as well as Sanger's. This is because in the examination, if there is, you might have to do it by yourselves. So we'll practice it in one of the classes in the coming classes. So you can read this is the template. The top strand is the template, and the bottom strand is the primer. Whatever we are reading on the bottom strand is actually that of the primer. If you want the template, and the primer is from 5' to 3'. If you want the template of this, then you should make a complementary of it. C, T, A, G, G, C, T, A, G, C. Like this, you can make, and you will then get the sequence of the template. And then you have to have, for template, 5' to 3'. Then you have the DNA that you are actually, then you have the sequence of the DNA you're trying to look at. Okay? Sanger's method is preferred over Maxman-Gilbert method for one particular reason. It is nice, it's easy to handle, and you can do you can sequence more number of bases per reaction. If you're running one maximum Gilbert method, you might know about 50 or 60 bases in one go. Whereas if you're doing Sanger's method, you can probably get 200, 300 bases also. Sometimes the gels were used to be very long sequencing gels, sometimes up to 2 meters or 1.5 meter maybe. People used to climb put a ladder and then load them up so that they can read larger sequences. That was the idea at that time. But the important important advantage of Sanger's method is it is easier for automation. I have been lucky to do Sanger's method, and this is how typically it would look like. Okay, the middle one is the sequencing reaction. As I was talking about the advantage of Sanger's method, and in Sanger's method, the advantage is it can be automated. It is much more easier to automate Sanger's method compared to that of um, maximum Gilbert method. In this case, they would have used fluorescent dyes, each colored differently. T could be in red, adenine in green, and so on. And you do only one reaction. You have all the components required for PCR. You would also have dideoxy ATP with the fluoro label. Okay, I will call it as what was there? Green. And dideoxy TTP with one color. So here it is one single reaction, right? Because the discrimination can be made based on the colors here. Dideoxy CTP, dideoxy GTP. Each of these dideoxy nucleotides are labeled with a fluorescent dye. It can be green, T is red, G is orange, and I think C is purple or so. That's not the color exactly, but for understanding purpose, I think blue is good. So they would have performed PCR reaction. So there would have been different sizes of fragments, each terminating with one of these. If that molecule had green color, that means the end of that molecule is A. If a molecule has red as is it fluorescing red, then it must be T. So then all these fragments are allowed to go through a fine capillary of capillary gel electrophoresis. It's a fine capillary, and the sample is loaded, and it will travel through this based on the size. And here we will have a laser beam. The laser beam will excite the fluorophore, which would emit the fluorescence, and that is detected by a detector. And that detector says if it is red color, it will automatically give it as A. If it is green color, it would give, sorry, if it is green color, it would say it as A. If it is red, it is T, and so on. So the computer will build a chromatogram, you can think of it like that, which will tell about the intensity as well. And then about other, sometimes you may have contaminating molecules, which may also show up. But the highest peak is what is the program will take it as the base in that location. Using this dideoxy or automated sequencing, the Sanger's based on Sanger's method, about some 400 to 800 or so, or 600 base pairs can be read per reaction. Even now, Sanger's method is used when people want to do small scale evaluations in some of their gene. Then they go for automated Sanger's method. But nowadays we have NGS. I think it is not included in your syllabus, but that by itself is a big course because it has to do with a lot of information technology, how you process the data, and so on. But I think if you are interested, you must have a look at the principles of NGS. NGS is the best one, but it is still expensive. And the problem, or that's not a problem. Basically, it is for large genomes, and so on. Large scale DNA sequences, then NGS is good to use. But if one is trying to do small, I just want to read about 1 kb, then people would go for Sanger's method. I'll stop with this one last, called as primer walking, which is nothing, it is similar to that of chromosome walking. In chromosome walking, we have, okay, sorry, I think I'll, 1 second, just be online. Can someone let me know if you still see the my screen?
Yes, sir.
Can you now see the whiteboard? Oh, okay. Can you see
Yes,
the
sir.
whiteboard?
Yes, sir.
This, yeah, okay, okay, yes. So in chromosome walking, we have done something like we tried to, the objective there was to find the order of genes. A Whatever gene is present, we wanted to find the order of genes on a gene, on the chromosome. In that case, we would have done gene library and tried to, using southern blotting, we would have figured out, or colony hybridization, we would have figured out which colony has a gene and which colony has the next gene of it, something like that. The order of genes is what we are trying to find. Very similar technique is primer walking, where we know, we know a small sequence, but we don't know the rest of the sequence on the genome. Then what we do is we would have designed a primer, and we will sequence this part, this much part, say some 600 base pairs or bases. Now I know this sequence, so I can design a primer based on that and use the primer to read through the next 600 bases. Like that, we continue going until we finish the sequencing the whole DNA fragment. Okay, that is called as primer walking. So I'll stop here and see if you have any questions.
Sir,
Yes, please.
sir, can you please explain the band to how we take